Welcome to the second part of our lecture on pharmacodynamics. This presentation will give an introduction to the concept of the drug-receptor interaction, the definitions of the drug receptors, ligands, as well as the different categories of the receptors, the properties of the receptors, such as the spare receptors and the silent receptors. We are going to start our discussion with the introduction to the drug-receptor interaction. So the interaction between a drug and the receptor produces the drug-mediated effects, which is an example of a cellular mechanism of drug action. Drug action is said to be specific. When we say specific, that refers to the interaction between the drug and the receptor. So in this case, the free and bound drug molecules that get distributed to the site of action will bind to the receptors. This resembles the lock and key relationship. This diagram shows the Fisher's lock and key hypothesis. In this hypothesis, it says that every lock has its own key. If the key is not precise, the lock does not open. The drug in this context represents the key that has to fit the target specifically and productively. So in this case, the lock represents the receptor and the key represents the drug. We also have another type of drug interaction in pharmacodynamics, and that is known as the non-specific interaction. So what is this non-specific interaction? So this is a kind of interaction that happens between a drug molecule and the plasma protein, particularly albumin. So this relationship or this interaction produces no perceptible drug effect. How do we define receptors? So when we say receptors, we basically define it as proteins. So most of them are proteins. They can either be located inside a cell or on its surface, which receives a signal. So this diagram shows the interaction between a drug to its receptor. So in this case, the drug represents the signal, and we have here the receptors that are found in the receptors that are found in the cell membrane, and this is the intracellular area of the cell. So we have here several drugs that are located in the extracellular space, and take note that only those drugs that are complementary to the receptors will bind to its receptors. So in this case, the drug, because it is complementary in shape to its receptor, it is specific to its receptor, so it will bind to that particular receptor to produce a particular effect. Another definition of the drug receptor is that it is a macromolecular component of the body tissue with which the drug interacts to initiate its pharmacologic effects. So these are molecules to which a drug has specific affinity. So in this case, this particular drug here has a specific affinity for this receptor because again, they have the specific complementary shape, and it also has the affinity to this particular type of receptor. So other types of receptor, or other types of drugs, because they don't have a complementary structure to this receptor, they don't have also the affinity to this particular receptor. So upon the binding of the drug to its receptor, that is known as the drug action, that is the time that the drug receptor complex will be formed, and that will produce a cascade of events that will eventually lead to the formation of the drug effects. So any drug or substance with specific affinity to receptors is called a ligand. The drug receptor has a ligand binding site as well as an effector site. So this diagram here represents an example of a representation of the ligand as well as the membrane receptors. So these are our membrane receptors. We also have here the ligands that are located in the extracellular space, and we also have here the intracellular space. So this is an example of ligand. The ligands are located outside the cell, and it will bind to the specific ligand will bind to its receptor based on the complementary structures in its shape as well as its specific affinity. So when the drug, for example, as a ligand will bind to its receptor, there is a time that the drug will the DR complex will form and the drug action will be exhibited. So upon binding, that is the time that the receptor will be activated and activating the receptor and forming the cascade of events that will eventually lead to the formation of the drug effect. The diagram shows a receptor that is located in the cell membrane. It is also known as a membrane receptor. It has three major parts: the ligand binding domain, the transmembrane domain, and the effector domain. The ligand binding domain is the part that is located in the extracellular part of the cell. The transmembrane domain is the one that spans the membrane, and the effector domain is the one that is located intracellularly. The ligand binding domain is the one that will interact with the ligand to form a receptor-ligand complex. Binding of a ligand to receptor will change its shape or activity, allowing it to transmit, signal, or directly produce a change inside of the cell conformation, inside of the cell. So as you can see here, there is a change in the conformation of the effector domain upon the formation of the receptor-ligand complex. So this change in the conformation of the effector domain will induce downstream effects, or it will induce some events that are necessary for the production of the biological response of this particular ligand. This is another diagram of a membrane receptor that is located on the cell membrane. So this is the extracellular fluid, the cell membrane, and the intracellular fluid. So the ligand will bind particularly to the membrane receptor, specifically on the ligand binding site of that particular receptor. So as you can see here, the ligand fits well with the ligand binding domain of the receptor. So upon the binding of the ligand to its membrane receptor, a ligand receptor complex will be formed, and this will trigger the intracellular responses for this particular complex. So the ligands can be classified as either endogenous or exogenous. So when we say endogenous, now these are substances that are produced or that originate within the living system. So examples of this are neurotransmitters. Examples of neurotransmitters are acetylcholine and epinephrine. We also have hormones. So example of hormones are insulin, glucagon, estrogen, and others. We also have the exogenous ligands. So when we say exogenous, these are those substances that originate from the outside of the living system. So examples of this are drugs and xenobiotics. So when we say xenobiotics, these are substances that are foreign to the body. What are examples of xenobiotics? So drugs is a xenobiotic. We also have the food additives as well as the pollutants, the environmental pollutants. Most of the drug receptors are considered to be protein in nature, and the receptors can also be present on the cell surface. Majority of the receptors are located on the cell membrane, and we also have other receptors that are intracellular. Now, they are also called as intracellular receptors. So these are located in the cytoplasm. So these are also known as cytosolic receptors, and we also have those that are found in the nucleus, also known as the nuclear receptors. This diagram shows the interrelationship between the ligands. We also have the membrane receptors and the secondary messengers. So these are important components of the drug action as well as the drug effect. So in this diagram, the ligands now are located extracellularly, so they are located in the extracellular fluid. We also have the receptors that are located on the membrane. So of course, these are known as membrane receptors. So typically, the receptors are made up of proteins, and again, they are embedded in the cell membrane. In this particular example, although there are many different ligands located outside the cell, the membrane proteins are specific, and only certain ligands will bind to each one of them. That is why each protein has a different ligand and also induces a different cellular response. So for example, in this diagram, this particular receptor has a specific ligand, based of course on its affinity and based on its the compatibility in terms of its structure. Also here, it has also a specific type of ligand, which is specific for its particular nature, and also in this particular receptor. Another component of this is the presence of the second messenger. So for example, when this ligand, specific ligand, will bind to its target receptor, so it will induce the formation of the secondary messengers. So what are these secondary messengers? So these secondary messengers are molecules. They are actually signaling molecules. They relay signals received at the receptors on the cell surface to target molecules particularly located in the cytosol or in the nucleus. Another important function of the secondary messenger is that they will serve to amplify the strength of the signal. Binding of a ligand to its receptor, particularly to a single receptor at the cell surface, may end up causing massive changes in the biochemical activities within the cell. So what are examples of the secondary messengers? There are three major classes of secondary messengers. Now we have the cyclic nucleotides. Example of the cyclic nucleotides are the CAMP and the CGMP. We also have the inositol triphosphate or the IP3, and the AG or the diacylglycerol. And another example is the calcium ions. So the whole event from the formation of the drug receptor complex up to the time that a signal will be relayed or will be amplified inside the cell in order to transmit that particular signal to be able to produce a biological response is an example of the signal transduction cascade or the signal transduction pathway. So the signal transduction, so basically when we say signal transduction, this is the transmission of the molecular signals from a cell's exterior to its interior. So this is only applicable for those drugs, those ligands that bind to the cell membrane. The signals in this particular event are received by the cells, and they must be transmitted effectively into the cell to ensure an appropriate response. So for example, a drug or particular ligand to produce a biological effect, the signal that is being produced from the binding of the ligand to its receptor must be transduced, or it must be amplified in order for the cell to respond effectively. So again, this step is initiated by the cell surface receptors. So for example, in this diagram, for when a ligand will bind to its specific receptor or target receptor, this will form a complex, and this complex will initiate a cascade of events that will lead to the formation of the secondary messengers. The secondary messengers are important signals that are being transmitted inside the cell to activate certain parts of the cell in order to ensure an appropriate response to be produced. So this is a diagram showing the step-by-step process in the signal transduction cascade or the signal transduction pathway. So we have here the presence of the receptor in the cell membrane as well as the ligand. So binding, so for example, when a ligand will bind to the cell surface receptor, what will happen is the receptor's intracellular domain changes in some way. So again, there is this is what we call conformational change within for example, the effector domain of that particular receptor. So generally, it takes a new shape, which may make it active as an enzyme or let it bind to other molecules. So that is the first step in the signal transduction cascade. So we have here the presence of the chemical messengers that are important important not to relay signals and to produce the biological response. This diagram shows the first step or the initial steps in the signal transduction pathway. So of course, the pathway is off when there is no ligand. When in the presence of a ligand, it will bind to its specific receptor, and it will also induce the binding will induce no conformational change, and it will activate that particular receptor. So the ligand, no, upon activation, will also activate the other proteins that are located at the cell membrane. So after that, the membrane protein will activate another protein that is located in the cytosol through the presence, of course, of the secondary messengers. Then, the protein in the cytosol will activate the final target of pathway, no, through, again, the presence of the secondary messengers. After that, the final target protein will cause the specific response. So this is, no, the summary of the signal transduction pathway. And of course, the secondary messengers are very important in order to amplify the signals that comes now from the binding of the receptor to its ligand in order to produce a particular biological response that is specific for that particular receptor and for that particular ligand. In terms of its chemical nature, the drug receptors could either be a protein, an enzyme, so mostly these enzymes are also proteins, and it can also be nucleic acids, and other cell constituents can also serve as drug receptors. So these are examples of the protein hormones, or rather protein receptors. Example is the muscarinic receptor that is located on the heart, the smooth muscle, or the exocrine glands. Another is the nicotinic receptors that are located at the neuromuscular junction. So again, for the protein receptors, example of that are the muscarinic receptors. So when we say muscarinic receptors, these are membrane receptors that are classified as G coupled protein receptors because again, the G proteins are important component of this type of receptors. They are involved in the parasympathetic nervous system. So the other term or the other name of this are muscarinic acetylcholine receptors because again the main ligand of this particular receptor are the acetylcholine neurotransmitters. So the other name of this particular receptor are 7 transmembrane receptors again because they span the membrane 7 times. We also have here the effectors, the key effectors for the muscarinic receptors. So these are chemical signals or chemical substances that are important for the production of the biological response of the acetylcholine and the muscarinic receptors. So another drug target are the enzymes, particularly the enzyme link receptors. So they have intrinsic enzymatic activity or they are associated with an enzyme, usually a kinase enzyme. So these are the three major structures or the key domains of the enzyme link receptor. The one that is located on the extracellular side is the ligand binding domain. We also have the transmembrane domain and the intracellular domain or the active domain. So for the enzyme link receptor, so the enzyme that is usually associated with this type of receptor are the kinases. So when we say kinases, now these are enzymes that catalyze the phosphorylation of the target molecules to cause their activation. So when we say phosphorylate, that is of course a word that that means to add a phosphate group to a molecule, a protein, or other kinase. So when there is the phosphorylation of a particular molecule, so it makes the particular molecule or that particular substrate to be activated. The opposite is true for the action of another enzyme, the phosphatase. So the action of this particular enzyme will inactivate that particular molecule or that particular substrate. So for the phosphatase, now another enzyme associated with this receptor, the phosphatase, these enzymes are necessary to dephosphorylate the target molecules So this dephosphorylation will usually inactivate the target molecule or the effector. So of course, no, yung phosphatase, they have an opposite action to that of the kinases. Example of the enzyme link receptor is the insulin receptors. So as shown in the figure. So binding of the ligand, which is insulin, to the insulin receptor will stimulate the carbohydrate utilization as well as the protein synthesis. We also have the physiological receptors. So these normally serve as receptors for endogenous regulatory compounds such as hormones and neurotransmitters. So of course, when we say hormones, the examples of this are insulin, glucagon, estrogen, and they are of course produced by the endocrine system. For the neurotransmitters, example of this are norepinephrine, epinephrine, dopamine, and serotonin, and they are produced by the nervous system. The cellular receptors can be broadly categorized into four families. From A to D, we have here the ligand-gated ion channel. Example of the ligand-gated ion channel is the nicotinic receptor or the cholinergic nicotinic receptor. We also have the G-protein coupled receptor. So examples of this are the alpha and the beta adrenoceptors. Another is the enzyme link receptors. So example of an enzyme link receptor is the insulin receptors. We also have another type of receptor that is located within the cell, and these are known as the intracellular receptors. So the intracellular receptors could either be located in the cytosol or in the nucleus. And example of this are the steroid receptors. So these are the four main types of receptors, now with their effectors and their location. So these three, the ligand-gated ion channel, the G-protein coupled receptors, and the kinase link receptors are located in the cell membrane. The nuclear receptors are, of course, a type of an intracellular receptor. In terms of their effector, the ligand-gated ion channels, its effector are the ion channels. No? So when we say effectors, these are molecules that translate the drug receptor interaction into a change in the cellular activity. For the G-protein coupled receptor, of course, its effector are the channels or the enzyme. And for the kinase link receptor, its effector is the enzyme kinase. And for the nuclear receptors, its effector is the gene transcription. So in terms of the examples, other examples of the ligand-gated ion channels are the gamma aminobutyric acid type. For the G-protein coupled receptor, so again we have the muscarinic receptors and the adrenoceptors. For the kinase link receptors, aside from insulin receptor, we also have the growth factor and the cytokine receptors. And for the nuclear receptors, so aside from the steroid receptors, we also have the thyroid hormone receptors. So let's start with the intracellular receptors. So again, we have here the outside of the cell, the membrane, and the inside of the cell. And this type of receptor is located in the cytosol. So for the intracellular receptors, so these are protein receptors that require the drug to cross the plasma membrane. So of course, the drug needs to be lipophilic. And another type is the transmembrane receptor. So for the transmembrane receptor, a drug will bind to the extracellular component of that particular receptor, and this will activate an enzymatic reaction into the intracellular component. Another type of receptor is the enzyme link receptor or the tyrosine kinase receptor. So when a drug will bind to this particular receptor in the extracellular component, this will lead to the dimerization of the two parts of the receptor intracellularly. So this dimerization will activate the tyrosine kinase, thereby leading to the phosphorylation of the tyrosine molecules on the target proteins. So another example of the receptor is the ligand-gated ion channel. So these ion channels are ligand-gated, meaning that they are closed until the receptor binds to the drug, which allows specific ions to pass by. So for example, drugs that stimulate the GABA receptors on the neurons causes the influx of the chloride ions. We also have another family of the cellular receptors are the G protein coupled receptor. So the G protein coupled receptor are similar to the tyrosine kinase receptor, and the drug receptor complex will lead to the interaction of the G protein with the receptor. This activated G protein will lead to the desired pharmacological response through one or a series of effector molecules or secondary messengers. The G protein coupled receptors are common types of receptors in the body. So majority of the receptors in the body are considered to be G protein coupled receptors. So we also have the spare receptors. So these are receptors that exist in excess of those required to produce a full effect. This applies to the situation when maximal response is elicited by occupancy of a fraction of the tissue receptors. So this means that not all receptors are needed for maximum response. So this represents the concept of the spare receptors. The x-axis represents the dose of the drug, and the y-axis represents the percent of maximum binding as well as the percent efficacy. So spare receptors are said to exist if maximal effect or maximal drug response is obtained at less than maximal occupation of the receptors. In practice, this determination is usually made by comparing two factors. So the first factor that we are going to compare is the EC50 that represents the concentration for 50% of the maximal effect with the concentration for 50% of the maximal binding or the KD. So if the EC50 is less than the value of the KD, then the spare receptors are said to exist. So in this case, our EC50 or the 50% maximal effect is achieved at a dose of 1.0, and our 50% maximal binding is achieved at a dose of 10. So our EC50 is less than our KD. So this just shows that a spare receptor exists. This is another representation of the concept of the spare receptor, showing the in the x-axis we have here the hormone concentration or the drug concentration. In the y-axis on the left we have the specific binding or percent of maximum, and on the right we have the biological response that is in percent of the maximum. So as you can see here, the ED50 for this particular hormone is achieved at this particular dose. So the ED50 represents the 50% of the maximal effect. The KD, or the 50% maximal binding, is achieved at 1 at this particular dose. So as you can see here, our ED50 is less than our KD. So we can say that in this particular hormone, the spare receptor exists. So this is the proportion of the spare receptor for this particular drug. So there is actually this particular amount of the drug receptors that are still unoccupied. So as you can see here, from this particular graph, the maximal effect of this particular hormone is achieved at this particular dose. So at a dose of 1, maximal effect of the drug is already achieved. So meaning that the proportion of the spare receptors are represented in this particular line here. So 50% of the receptors are still unoccupied, and the drug has already achieved its 100% or its maximum effect. So at this particular portion here of the graph, 50% maximal effect has been achieved at about 30% binding. At 50% binding, 50% maximal binding, 100% of the biological response has already been achieved. So therefore, the other 50% of the receptors are still unoccupied. So in this example, the spare receptors exist. So this is another representation of the spare receptor concept, with the y-axis representing the percent receptor occupancy, and the y-axis represents the effect of this particular drug. So again, on the x-axis, we have the percent receptor occupancy. So in this particular line here, the line graph, there is a linear relationship. Now, 50% of the total receptor population must be occupied to achieve 50% of the maximal organ response, or a maximal effect, or the maximal organ response. So as you can see here, in this linear relationship, there is a 1-to-1 ratio or proportion. No? So meaning that for every 50% of the receptors that has been occupied, there is also a 50% maximum effect that is achieved. Conversely, at 100% occupancy of the receptor, there is also a 100% maximum effect. So this type of linear relationship does not exhibit a spare receptor concept, because all of the receptors must be occupied in order for this particular drug to achieve its maximum effect. For this particular drug here, there is a nonlinear relationship. At 50% receptor occupancy, you can see here that it has already achieved its maximum effect. So therefore, there is still 50% of the receptors that are unoccupied. So this particular drug exhibits a spare receptor. So let's now proceed to the concept of the acceptor and the silent interactions. So how do we define silent receptors? So these are sites which bind specific drugs, but no pharmacological response is elicited. So they are better called as drug acceptors. Examples of this are plasma proteins, which have binding sites for many drugs. Example of the plasma protein is the HSA, or the human serum albumin. That is the major plasma protein in the blood. So to avoid confusion, some references have recommended to use the term receptors, that the usage of the term receptor should be restricted to those regulatory binding sites which can generate a response. So in the case of the silent receptors, such as the albumin, some references recommend them to be called drug acceptors rather than as receptors. What is the significance of the binding of the drug to plasma proteins? So the binding of the drugs to plasma as well as tissue proteins has significant consequences on both the PK and the PD of the drug, because only the free fraction of the drug can freely diffuse across the membrane to different tissues of the body. So for example, when you are going to administer a particular drug, so orally, what will happen is it will of course undergo, it will pass through the stomach, it will undergo the first pass effect, and after that it will be absorbed in the bloodstream. So in the blood, the drug can be available in its free, unbound form, and also its bound form. So when we say bound form of the drug, these are the fraction of the drug that are bound to proteins, plasma proteins such as albumin. And when we say free form of the drug, ito yung mga unbound form of the drug. So those fraction of the drug that are free would be able to readily cross the plasma membrane, now provided of course that they are lipophilic. They are free. No? They are able to cross the plasma membrane, and they are also considered to be pharmacologically active as compared to those drugs that are bound. So yung mga bound form of the drug, they cannot be able to readily pass through the plasma membrane, so they are pharmacologically inactive.